everyone. Welcome to the microbiology sessions. Today we will be talking about the morphology of bacteria. Before we go into the actual anatomy of bacteria, let us talk a little bit about the introduction. You know about the animal kingdom and the plant kingdom. Bacteria belongs to the Protista kingdom, neither of these two. Protista kingdom consists of two groups, the prokaryotes and the eukaryotes. Prokaryotes are usually unicellular organisms without any branchings, whereas the eukaryotes are multicellular and distinct. The eukaryotes, as you have already read in schools, etc., they all come with the nucleus, nuclear membrane, nucleolus, endoplasmic reticulum, mitochondria, Golgi apparatus, etc. But in case of prokaryotes, all these are absent. What they have are distinct structures for all the essential functions like reproduction and excretion, etc., and energy production. These different structures we are going to be dealing with in today's chapter. So, these are all the different structures of the anatomy of the bacterial cell, which you are going to be studying about today. So, let us first come to the cell wall. What is a cell wall? Cell wall is a very rigid structure which is present all around the bacterial cell. It gives the shape and the size to the bacterial cell. Now, because it is rigid, it gives protection also from the osmotic damage or from antibiotics or lysosomes or any other actions which can affect the bacterial cells. Also, it has a certain antigenicity, so it is capable of producing antibodies in us. So, how does the structure of the cell wall look like? The cell wall is basically made up of a peptidoglycan layer. This peptidoglycan layer consists of two different types of polypeptides or polymers. The first one is N-acetylmuramic acid or NAM, and the other one is N-acetylglucosamine or NAG. Both NAM and NAG are present alternate to each other in conjunction with each other, forming a very strong net-like structure. So, basically they are called scaffolds and they look like the net of a tea filter or anything like that. Okay. So, this scaffolding basically it is so strong that it is difficult to come off. So, that is what gives the strength of the cell wall to the bacteria. And the glucosamine also or the NAG also consists of 4 different tetrapeptide links, and these tetrapeptide links of each of the NAG are connected with a pentaglycine cross-link. All these structures together give a strength to the cell wall so that nothing can pass through or affect the bacterial cell. So, we have discussed about gram-positive and gram-negative bacterial cell. Now, the gram-positive and the gram-negative nature of the bacterial cell actually comes from the structure of the cell wall. So, in other words, the cell wall structure of a gram-positive is different from that of the gram-negative. So, what is the difference? This peptidoglycan layer which is present, that is very, very thick in case of a gram-positive cell wall. In case of gram-negative cell wall, the same peptidoglycan layer, the structure is the same, but the thickness is not as much as a gram-positive cell wall, it is very thin. But of course, we will come to that a little later because it has other structures also present on it. Now, in case of a gram-positive cell wall, other than this thick peptidoglycan layer, there are other structures which are called teichoic acid, which also gives strength to the bacterial cell. Now, this teichoic acid, there are two ways in which they come up. They are either from the cell membrane itself, where they are called as membranous teichoic acid, or they come from the cell wall itself, the peptidoglycan layer. So, they are called the cell wall teichoic acid. Okay. Now, this also gives further strength to the bacterial cell. In case of a gram-negative bacterial cell, I told you the peptidoglycan layer is very thin. So, just because it is thin doesn't mean that it doesn't give, there is not much of protection to this cell. The protection is there because outside this cell, as you can see, there is a very thick outer membranous layer. Now, this outer membranous layer is a lipoprotein layer, starting with a lipoprotein layer which basically connects the peptidoglycan layer to the lipid layer on the top. This layer consists of a number of structures called porins, which is basically made up of a protein A. Apart from porins, there are a lot of other proteins also, which are also called protein A. Now, this protein porins are basically diffusion channels, which are for small particles to go in and out also, and also for the bacteriophages to attach themselves.